So let's say we have two plasmids, pGex5x3 and plasmid TDL14, and the purpose of this experiment is to subclone a human thrombopoietin gene from TDL14 to pGex5x3 so that we can eventually express it as fusion protein. So let's open pGex5x3, which is our expression vector, as well as plasmid TDL14, which contains human thrombopoietin gene. So these are the two text panes which contain a lot of information about both these plasmids, and I'll now create graphic map of both these plasmids by going down at the bottom and clicking on graphic map of pGex5x3. I'll close the text panes and I'll closely inspect the graphic map so that I can devise a strategy to subclone human thrombopoietin gene from TDL14 to pGex5x3. So I just removed the unique sites, restriction sites, to see the plasmids more clearly. On the left side is TDL14, on the right side is pGex5x3, which is the expression vector. And in the TDL14 from 380 to 1442 we have human thrombopoietin gene. In pGex5x3 the whole expression cassette is driven by tag promoter, and at the end of it we have a multiple cloning site where we would like to subclone our genes. This is tag promoter, which will drive inducible expression of human thrombopoietin gene once we clone it. So this is the multiple cloning site where we intend to introduce human thrombopoietin gene. So let's visualize the unique sites or restriction enzymes that cut TDL14 singly to figure a strategy by which we can subclone human thrombopoietin gene. So it spans from 380 to 1442 base pairs. So obviously we need restriction enzymes which cut before the start of the gene, let's say BAMH1, which cuts at 371, or let's say NHE1, EcoR5. So we can choose one of those. Which will fall in that area, the blue area. And downstream, we can pick restriction enzyme that is listed after the gene ends. Right, so let's say 1442 SAL1. Or like XMA3. So you can pick enzymes which you already have in your lab or you can easily acquire. Now let's look at the unique restriction enzymes of pGex5x3. So the multiple cloning site goes from 934 to 971. At 934 we have BAMH1. All these enzymes, they're part of multiple cloning site. And we have a SAL1 at 954. So if you remember, we saw these two enzymes on both sides of human thrombopoietin gene also in TDL14. So BAMH1 cuts before human thrombopoietin gene and SAL1 cuts after, right? And so we can use BAMH1 and SAL1 because in the multiple cloning site of pGex5x3 again we have BAMH1 and SAL1. The key is that we need to remember, once we clone the gene, it should be in the right orientation, right? So let's open the construct window and open the cloning or expression vector pgex5x3. Remove unique sites and visualize BAM-H1 and SAL1 by using particular sites. From the drop down menu, select BAM-H1 and SAL1. So because we want to do inducible expression and we want our gene to be in the right orientation with respect to tag promoter, that's why we are doing directional cloning. So here we'll click on SAL1 first, and then in the clockwise direction we'll press shift, and then we'll click on BAM-H1. So it will select the longer fragment. We have as second sequence we have plasmid TDL14. So again we'll visualize the BAM-H1 and SAL1 from the particular site, which will be on both sides of human thrombopoietin gene. So this is why whenever we need to do expression of a gene, we have to do a directional cloning. So BAM-H1, and in the clockwise direction we'll press shift and then press SAL1, right? So it will cut and select the piece which contains human thrombopoietin gene. Then we press ligate, and there we go. We have the new construct. This is its graphic map. I'll just minimize. Okay, I'll close off the parent plasmids. Now let's compare our new construct, ligation number 1, with the parent plasmid pgex5x3, right? So I'll remove all the unique sites and visualize the BAM-H1 and SAL1. There we go. And same thing for the parent plasmids pgex5x3. There it is. It starts at 934, BAM-H1. So as you can see, 934 BAM-H1 starts. That's where the restriction site is, and that's where we have cloned human thrombopoietin gene as part of an insert from pTDL, and this is where it ends at SAL1. 4974 size of pgex5x3, 6025 size of our new construct. Increase in size proportional to the insert we added from pTDL14. So now we'll save our construct. Instead of ligation number 1, we can name it as, let's say, human thrombopoietin gene in pgex 3x. I'm sorry, it's pgex 5x3, but let's go with it for a while. So this is our new construct. I'll just digest it with BAM-H1 and SAL1. So this linearized the plasmid, and now with SAL1 we got two fragments, the larger one which is the backbone of the plasmid and which is 4954 base pairs. The shorter fragment 1071, that's what contains human thrombopoietin gene. So this fragment 1071 should also be there in plasmid TDL14, right? So let's do that. We'll take plasmid TDL14 and digest with BAM-H1 SAL1, and here we go. So we again got this 1071 band. So that confirms that we got the right fragment in our new construct.